Welcome to all students. Myself, Om Prakash Vodara, Assistant Professor, Crop Physiology, Department, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapits, Women's University, Jaipur. Today, we discuss about our topic photosynthetic pathway, C3, C4, and CM mechanism, plants, dark reactions or black reactions or path of carbon in photosynthesis. This is the second step in mechanism of photosynthesis. The chemical process of photosynthesis occurring independent of light is called dark reactions. Mainly जो light है वो mainly इसका role नहीं होता dark reactions के अंदर और ये independent होती है. It take place in the stomach of chloroplast. Mainly जो ये reactions है dark reactions वो chloroplast के stomach जो parts होता है उसके अंदर complete होती है. A dark reaction is purely enzymatic, mainly completely enzymatic reactions होती है dark reactions and it is slower than the light reactions. Light reactions की तुलना के अंदर इसकी जो process है वो slow होती है. The dark reactions occur also in the presence of light. ये जो light है उसकी presence में भी mainly कुछ part है इसका occur होता है. In dark reactions the sugar are synthesized from CO2. Mainly जो sugar है उसका जो synthesize है वो mainly CO2 से होता है. The energy poor CO2 is fixed to energy rich carbohydrate using the energy rich compounds. ATP and all asymmetry cause NADPS2 of light reactions. The process is called carbon fixation or carbon assimilation. Mainly जो light reactions है जिसके अंदर mainly ATP और NADPS2 का जो formations होता है वो mainly dark reactions के अंदर जो carbon fixations है या carbon assimilations है उसके अंदर mainly use होता है. Since Blackman demonstrate that accidents of dark reactions, the reactions is also called Blackman reactions. Mainly जो Blackman's firstly इन्होंने इस डार्क रिएक्शन को डिस्कवर किया था तो उसके अकॉर्डिंग इस रिएक्शन का जो नेम है वो Blackman's reactions के नाम से भी जान सकते हैं. In dark reactions, two type of cyclic reactions occur. Mainly जो dark reactions है इसके अंदर two types से जो cyclic reactions है वो mainly occur होती है Kelvin cycle और C3 cycles and Hatch and Slack pathway or C4 cycles. Kelvin cycle or C3 cycles, it is cycle reactions occurring in dark phase of photosynthesis. In these reactions, CO2 is converted into sugar and hence it is a process of carbon fixations. The Kelvin cycle was first observed by Melvin Kelvin in chlorella. एक प्लांट था जिसके अंदर मेलविन केल्विन साइंटिस्ट ने सबसे पहले जो केल्विन साइकिल से वो ऑब्जर्व किया. ये जो chlorella है वो एक यूनिसेल्युलर ग्रीन एल्गी से होती है. Kelvin was awarded a Nobel Prizes for this work in 1961. Since the first stable compounds in Kelvin cycle, three carbon compounds, three phosphoglycic acids. So the cycle is also called as C3 cycles. The reactions of Kelvin cycle occur in three phases. Mainly occur in three phases: carboxylative phase, reactive phase, and regenerative phase. तो different जो phases है उसके अंदर ये mainly complete होती है. Carboxylative phase, three molecules of CO2 are accepted by three molecules of five carbon compounds which ribose bisphosphate to form three molecules of an unstable intermediate six carbon compounds. These reactions is catalyzed by the enzymes carboxy dismutase. Mainly जो 3CO2 है molecules होते हैं उनका जो mainly interactions है वो 3 ribose disphosphates mainly carboxylase dismutase enzyme की उपस्थिति में इनका जो convergence है वो 3 unstable intermediate 6 carbon compounds के अंदर होता है. The three molecules of unstable 6 carbon compounds are converted by the additions of 3 molecules of water into 6 molecules of 3 phosphoglycic acid. This reaction is also catalyzed by enzyme carboxymutase and after that the formations of 3 phosphoglycic acid. 3 phosphoglycic acid is the first stable product of dark reactions of photosynthesis. तो many dark reactions है उसका जो first stable product है वो 3 phosphoglycic acid होता है and since it is a 3 carbon compound this cycle is known as C3 cycles. Second, reductive phase, 6 molecules of 3 phosphoglycic acid are phosphorylated by 6 molecules of ATP produced in the light reactions to yield 6 molecules of 1,3-diphosphoglycic acid and 6 molecules of ADP. After that, mainly it 1,3-diphosphoglycic acid conversions into interactions with Six molecules of 1,3-diphosphorylic acid are reduced with the use of six molecules of NADPS2 produced in light reactions to form six molecules of 3-phosphoglyceraldehyde. This reaction is catalyzed by the enzymes triose phosphate dehydrogenase. After that, regenerative phase start. In the regenerative phase, the ribose bisphosphate is regenerated. The regenerative phase is called as pentose phosphate pathway or hexose monophosphate shunt. It involves the following step. Some of the molecules of 3-phosphoglyceraldehyde into dihydroxyacetone phosphate, both 3-phosphoglyceraldehyde and dihydroxyacetone phosphate, then unite in the presence of the enzymes aldolase to form fructose 1,6 bisphosphate. After that, fructose 6 phosphates is converted into fructose 6 phosphate in the presence of phospholipase enzymes. And some of the molecules of 3-phosphoglyceraldehyde instead of forming hexose sugars are diverted to regenerate ribose 1,5-diphosphate. 3-phosphoglyceride react with fructose 6 phosphates in the presence of enzyme translocase form erythrose 4 phosphates 4 carbon sugars and cellulose 5 phosphates 5 carbon sugars. Erythrose 4 phosphates combines with dihydroxyacetone phosphates in the presence of the enzymes aldolase of form pseudoheptulase 1,7 and diphosphates 7 carbon sugars. Pseudoheptulase 1,7 diphosphates loses one phosphate group in the presence of the enzymes phosphatases to form pseudoheptulase 7 phosphates. Pseudoheptulase phosphate react with the 3-phosphoglyceraldehyde in the presence of transketolase enzymes to form cellulose 5-phosphate and ribose 5-phosphates. Both the percentage of sugars are equals. Ribose 5-phosphates is converted into ribose 1,5,5 bisphosphate in the presence of enzymes phosphopentokinase and ATP. Two molecules of xylose phosphates are also converted into one molecule of ribose monophosphate. The ribose monophosphate is phosphorylated by ATP from ribose bisphosphate and ADP. Ribose mainly converted in dark reactions. CO2 is fixed carbohydrates and the CO2 acceptor ribose diphosphates is regenerated in Kelvin cycles. And 12 NADPS2 and 18 ATP are required for fixed 6 molecules into one hexose sugars molecules, fructose 6 phosphate. After that, mainly light reactions in which mainly photosystem seconds electron transport chains photosystems mainly ATP and NADPS convert to NADPS2 and energy is produced. C4 cycles or Hatch and Slack pathway. It is the alternate pathway of C3 cycles to fix CO2. In these cycles, the first formed stable compound is 4 carbon compound with oxaloacetic acid. Hence, it is called C4 cycles. The pathway is also called as Hatch and Slack as worked out the pathway in 1966, and it is also called as C4 dicarboxylic acid pathway. This pathway is commonly seen in many grasses, sugarcane, maize, sorghum, and amaranthus. The C4 plants show a different type of leaf anatomy. The chloroplasts are dimorphic in nature. The leaves of these plants, the vascular bundles, are surrounded by bundle sheath of larger parenchymatous cells. These bundle sheath cells have chloroplast. These chloroplast of bundle sheath cells are larger, lay grana, and contain starch grains. The chloroplast in mesophyll cells are smaller and always contain grana. The peculiar anatomy of leaves of C4 plant is called cranch anatomy. The bundle seed cells are bigger and look like a ring or worth. Cranch in German means worth and hence it is called cranch anatomy. The C4 cycle involved two carboxylation reactions, one taking place in chloroplast, mesophyll cells, and another in chloroplast of bundle seed cells. There are four steps in Hatch and Slack cycle. First, carboxylations, second, breakdowns, and third, splitting, and fourth, phosphorylations. First, carboxylations. It takes place in chloroplast or measurable cells. Phosphoenol pyruvate has three carbon compounds which pick up CO2 and change into four carbon oxaloacetic acid in the presence of water. This reaction is catalyzed by the enzyme phosphoenol pyruvate carboxylase. Certain breakdowns, oxaloacetic acid break down readily into four carbon malates and aspartate in the presence of the enzymes transaminase and malate dehydrogenase. After that, splitting is starts in the sheath cells. Malate and aspartate split enzymatically to yield free CO2 and three carbon pyruvates. The CO2 is used in Kelvin cycles in sheath cell. The second carboxylation occur in the chloroplast of bundle sheath cells. The CO2 is accepted by five carbon compounds, ribulose diphosphates in the presence of enzyme carboxylase dismutase and ultimately yield 3-phosphoglycic acid. Some of the 3-phosphoglycic acid is utilized in the formations of sugar and the rest regenerate ribulose diphosphate. After that, last procedure phosphorylations. The pyruvate molecules is transferred to chloroplast of mesophyll cells where it is phosphorylated to regenerate phosphoenol pyruvate in the presence of ATP. This reaction is catalyzed by pyruvate phosphokinase and the phosphoenol pyruvate is regenerated. In H and Slack pathway, the C3 and C4 cycles of carboxylation are linked and is due to the crank anatomy of leaves. The C4 plants are more efficient in photosynthesis than C3 plants. The enzyme phosphoenol pyruvate carboxylase of the C4 cycles is found to have more affinity for CO2 than the ribulose diphosphate carboxylase of C3 cycles in fixing the molecular CO2 in organic compound during carboxylations. Crystallization acid metabolism cycles or the dark fixation of CO2 in succulent plants. CM is a cycle reaction occurring in the dark phase of photosynthesis in the plant of crystallization. It is a CO2 fixation process wherein the first product is malic acid. It is the third alternate pathway of Calvin cycle occurring in mesophil cells. The plant exhibiting CM cycles are called CM plants. Most of the CM plants are succulents. Example, Bryophyllum Kalonji, Crassula, Pseudium, Glenia, act etc. also seen in certain plants, cactus, example, or Opuntia species, orchids, and pineapple families. CM plants are usually succulents and they grow under extremely zero, xerotic conditions. In these plants, the leaves are succulents or fleshy. The mesophyll cells have large number of chloroplasts and the vascular bundle are not surrounded by the well-defined bundle cells. In these plants, the stomata remains open during night and closed during daytime. The CM plants are adapted to photosynthesis and survival under adverse xerophytic conditions. CM plants are not an efficient as C4 plant in photosynthesis, but they are better suited to conditions for extreme desiccations. CM involve two-step acidifications and deacidifications. Firstly, acidifications in which mainly in darkness the stored carbohydrate are converted into phosphoenol pyruvic acids. The process of glycolysis. The stomata in CM plants are open in dark and they allow diffusions of CO2 from the atmosphere into leaf. Now, the phosphoenol pyruvic acid is carboxylated by the enzyme phosphoenol pyruvic and carboxylase and it converted into oxaloacetic acid. The oxaloacetic acid is then reduced to malic acids in the presence of enzyme malic dehydrogenase and the reactions require NADPS to produce in glycolysis. The malic acid produced in dark and stored in the vacuoles, the malic acid increase the acidity of the tissue. After that, de-acidifications occur during daytime. Mainly जो acidifications है वो इंक्रीज होता है और जो एसिडिफिकेशन है उसकी जो रिवर्स प्रोसेस है वो मेनली स्टार्ट होती है तो इस कंडीशन में मेनली जो प्लांट है वो जो एज लाइक जो C4 साइकिल्स है वो क्रिएट होता है इस प्रकार तो मेनली जो प्लांट है उसके अंदर जो फोटोसिंथेसिस मैकेनिज्म है वो डिफरेंट प्लांट के अंदर डिफरेंट तरीके से होती है। जैसे थ्री प्लांट है उसकी जो मैकेनिज्म डिफरेंस होती है, और C4 एंड CM प्लांट्स है उसके अंदर मेनली जो फोटोसिंथेसिस मैकेनिज्म है डिफरेंट पाई जाती है। तो अकॉर्डिंग टू सिचुएशन जो प्लांट है किस कैटेगरी है, तो वो डिफरेंट जो क्रॉप्स है वो, या जो प्लांट है आपका हर्ब्स है या जो झाड़ी है बसेज है, तो उसके अंदर मेनली जो फोटोसिंथेसिस मैकेनिज्म है वो डिफरेंट पाई जाती है। तो थैंक यू ऑल स्टूडेंट्स।